So it's my big pleasure to introduce today's speaker, Dr. Anne Medeal, who is a Florence McAllister Professor of Medicine at Duke University. She's a true physician scientist and clinical hepatologist. Her research activities are focusing on basic mechanism of different aspects in hepatology, liver repair, they complement, and not only liver repair, actually also cancer and fatty liver disease, and they complement the clinical and translational activities. She's been very well funded through the NIH with different R01s and U01s, and has provided significant contributions to the field. And I mentioned regeneration, alcohol, fatty liver disease. She's a member of the American Society of Clinical Investigation and the Association of Academic Professors. She has received an NIH NIAAA Merit Award, an NIAAA Mendelssohn Award, and the Duke Distinguished Faculty Award. In other words, and I don't want to take too much time because otherwise we won't get a chance to talk to her. And again, I'm super excited to listen to her talk, and the title is Liver D or Regeneration and Cancer. And the floor and camera is yours.
Thanks, Tim, for the kind introduction. And thank you, Jen, and everybody at NCI for inviting me to talk about some of my favorite topics. And I crossed out re because I'm going to try to convince you today that the liver diseases we're worried about are actually results of degeneration of the liver. So let me see if I can get this to move. Uh-oh, we're having a problem. There we go. So I don't know if many of you are aware, but there is some nomenclature changes that are coming down the road. And there's talk about lumping together fatty liver diseases, alcohol-related fatty liver disease, and NAFLD under a new category that they call steatotic liver diseases. And that's what I'm going to actually talk about today, and how they relate to liver cancer and to cirrhosis. But I'm going to try to convince you that this is justified because over the last few decades, the prevalence and incidence of viral hepatitis, particularly in the Western world, is decreasing now that we have effective vaccines and treatments. But the prevalence of the steatotic liver diseases, NAFLD, non-alcoholic fatty liver disease, and alcohol-related fatty liver disease are increasing. And so that it is these steatotic liver diseases that are now driving the increasing incidence and prevalence of cirrhosis and liver cancer. And in a way, this is fortunate because we actually know a fair amount about the biology of steatotic liver diseases. And we know that NAFLD and AFLD have two key similarities. Both are triggered by metabolic stress. Alcohol and food both deliver excessive calories. And I'm going to try to convince you today that both are degenerative processes. And in fact, if you think about it, they're both systemic diseases. And the liver isn't the only tissue that's affected. There's heart problems, there's pancreas problems, there's kidney problems, there's bone problems. And so that prompted me to go to my favorite source, which is Wikipedia, and look up degenerative diseases. And Wikipedia defines degenerative diseases as a result of a continuous process based on degenerative cell changes affecting tissues and organs, which will increasingly deteriorate over time. And I said, that kind of looks like this cartoon, which almost everybody who speaks about fatty liver disease uses, where we show a healthy liver frequently becoming steatotic. That's a reversible disease, but some people with steatosis get a more serious form of liver damage called steatohepatitis. Even that can be reversible in some people, but over time can progress to fibrosis, cirrhosis, and cancer. So if these are degenerative diseases, what is tissue degeneration? Well, it's caused by a loss of cellular resiliency. And in that sense, it's a maladaptive state in which the tissue becomes more vulnerable to injury and less able to recover. And so you get aberrant repair that causes scarring and cancer. In other words, wounds that don't heal. So with that as a background, we began to test the hypothesis that fatty liver diseases are related to that cause cirrhosis and cancer, actually result from liver degeneration. And we know that both of these conditions, cirrhosis and cancer, are largely restricted to the subgroup of people who develop actual injury in the organ or steatohepatitis. And then we've identified variables that seem to associate with a greater risk of developing fibrosis, cirrhosis, and eventually cancer. And these include age, male sex, diabetes, cigarette smoking. And so I'm going to just show you some data that sort of emphasize that point. This is a recent paper published in 2021 from Art McCullough's group at the Cleveland Clinic. And they looked at close to 400 NAFLD patients that they had seen there over a 5-year period, and they found about 1,000 or so had liver cancer. Notice that the chance of getting liver cancer was much less in people who were not cirrhotic than people who were cirrhotic. And I showed you before the risk factors for going from steatohepatitis to cirrhosis. And I want to show you that these same risk factors are associated with the likelihood of getting liver cancer, whether you have cirrhosis or not. So they include age, male gender, diabetes, if you're a smoker, if you have a higher ALT, in other words, if you have steatohepatitis. And at the top of the list for all of these is age. So age associates with tissue degeneration. So both cirrhosis and liver cancer are really two sides of the same coin. They are both consequences of liver degeneration. So with that as a handle, or a hook, maybe we can dive a little bit deeper in. So degenerative diseases, as I said earlier, are strongly associated with and exacerbated by aging. They cause premature mortality. And interestingly, the age-dependent component of mortality increases with chronological age. So to some, degenerative diseases are caused by accelerated or premature aging. And in other words, for accelerated or premature aging is your biological age is greater than your chronological age. So a long background, why should you care? Because aging is reversible. So I'm citing here one of a number of papers that have come out recently where people have actually been able to harvest material from young animals, in this case, extracellular vesicles, and give them to old animals, and they could revert some of the tissue aging phenotype of the old animals, in this case, sarcopenia. And some of our current steatotic liver disease treatments actually can reverse aging, or at least slow it down. So for the rest of the talk, I'm going to focus on biological aging and address what is it, what accelerates it, what delays it, and what difference does it make. So biological aging is loss of resiliency or loss of plasticity. So if you're young and a cell gets hit by a stress like alcohol or excessive caloric intake, the cell adapts and it survives that stress. And then when the stress is removed, it reverts back to where it was before. So young tissues have a lot of plasticity. And this is my 7-year-old granddaughter, Rosie. At the time she was 6, Rosie fell off the monkey bars, literally broke her arm in half, had to have surgery. And she looked pretty punk when she came back that evening, but by the next morning, Rosie was ready to go. So Rosie is resilient, Rosie is young. Biological aging is loss of resiliency or loss of plasticity. So a stressor comes and either the cell does not adapt or it adapts, but when the stress is removed, it can't snap back. So the tissue begins to accumulate cells that have adapted, but it's appropriate as long as the stress is there, but it's maladaptive if they stay that way and the stress is removed. So biological aging is loss of resiliency. What accelerates and what delays loss of resiliency? So the rate determinants of biological aging are genetic, epigenetic, and time or chronological age. So as we know, genetics is the hardware of our cells. It's the DNA code. And in the beginning of things, when we had the Human Genome Project, we were all hopeful that it would be very simple to figure out disease because it was clear. All you had to do was change the code. You could change the trait, and that would explain whether or not you had disease. And of course, we have identified major risk alleles for cirrhosis and cancer associated with steatotic liver disease. And I list some of them on this slide. And notice that they're all related to metabolism. They're all lipid-related genes. And we know that there's some sort of a interaction among these factors because the more of them that you have, the more likely you are to get cirrhosis or liver cancer. And this is actually percolated to prime time. As you well know, we're now doing clinical trials by trying to actually edit some of these genes and see if we can affect outcomes. So that's great, but what does fat in hepatocytes have to do with premature aging, liver degeneration, cirrhosis, or cancer? So I'm going to ask you to hold that thought because we're going to come back to it in a few minutes. But first, it's important to reflect that although these genes are strongly associated with progression or bad outcomes of steatotic liver disease, we know that they're not sufficient to drive that phenotype. Because if you look at the blue lines on these graphs here, they're the bad PNPLA3 genotype. And notice that you can have the bad genotype, but if you have a low BMI, you're not likely to have fat in your liver, an elevated ALT, or to get cirrhosis of the liver. So the risk for all of those things goes up as your BMI goes up. So in other words, it looks like there's some sort of an interaction between the genotype and potentially the external trigger for metabolic stress. So what controls the actual phenotype? And I think increasingly we're recognizing the importance of the epigenome, which is the dynamic maintenance system that regulates gene activity. In other words, whether the mutated or non-mutated gene is actually expressed. And one of the things that regulates that importantly is DNA methylation. And the reason I focus on that is because of this data now that we know about called the Horvath clock. And the Horvath clock is basically derivative of an observation that chronological age predictably alters the DNA methylome. Some of those changes are pre-programmed during development in utero. Others are thought to be cumulative consequences of life, and the latter is called epigenetic noise. Both of those seem to alter DNA methylation and therefore modify regulation of gene transcription. These global changes in gene expression that then result impact stress responses or resiliency. So environmental and genetic perturbations can increase or decrease the effects of chronological age. Your biological age can be younger than your chronological age, as shown on the blue dots, or it could be older than your chronological age, as shown in the red dots. And it would be people whose biological age is older than their chronological age that are having accelerated tissue aging. So what we talked about so far is that biological aging is loss of resiliency. It's regulated by genetics and epigenetics. What difference does it make? Well, this is a study that Rohit Lumba and I did a number of years ago where we looked at some of the patients that they had accumulated at UCSD that had NAFLD. And some of the patients had different stages of fibrosis. Some were controlled, some had mild fibrosis, some had more severe fibrosis. And we calculated the Horvath clock, the methylome in the blood of these patients. And what we were surprised to see is that the patients who had NASH had an older biological age than the patients who did not have NASH. So they are shown at the yellow things that are above the line here on this graph. And then if we looked within the cohort of people that had NASH, if we looked at the people who had more advanced fibrosis, you can see that the people with more advanced fibrosis tended to have an older biological age. So maybe everything that we talked about before does have some relevance to disease progression, at least in fatty liver disease. And this could be important because the things that we recommend to our patients with fatty liver disease are actually known to affect DNA methylation and therefore might reset the biological clock. So if all of this is true, then it becomes important to understand, is the liver the villain or the victim here? So in order to try to look at that at a little bit deeper level, we wanted to ask, does the liver burden of old cells correlate with NAFLD severity? So as we said before, degenerative diseases are caused by accelerated aging, which would mean that the affected tissues progressively accumulate cells with hallmarks of aging. These hallmarks of aging would identify old cells. And the more of those old cells you have, if our hypothesis is correct, you should have progressive deterioration of tissue function and more advanced stages of the disease. So the aging field has come up with a list of things that they call hallmarks of aging, that's shown on the right, where you can see genomic instability, telomere attrition, that whole circle of things. One of the things that they've identified is cellular senescence, and they have certain biomarkers of senescent cells, and they include expression of cell cycle inhibitors like P21 and P16, which would cause cellular senescence. So we decided to look for these hallmarks of aging in patients that were in the Duke NAFLD biorepository. This is a biorepository of a lot of people. We picked 368 who had had a diagnostic liver biopsy from which we had clinical data, liver histology, and some blood biomarkers. And what I'm trying to show you on this slide is just that our NAFLD cohort is enriched actually with people that have diabetes, which is one of the things that should accelerate aging. About half of them had diabetes. And then it's got a fair number of people that have advanced fibrosis, F3 or 4 fibrosis, about 30-some%. And then if you look at the NASH score, the vast majority of people in this biorepository have NASH. So the first thing we did was bulk RNA-seq, and this just shows you that the experiment worked. On the left-hand side, you see the group that has F3, 4 fibrosis. They have different genes that are up or down regulated compared to the controls. And some of the genes that are upregulated are the ones that you would expect that would go along with fibrosis. So here we looked at senescent signatures in our patients who had NAFLD and a control group of patients who did not have NAFLD. And on this slide over here, you can see that the group that had NAFLD were more likely to have this signature of senescence. And here are our biomarkers, P21 and P16, and you can see that they're increased in the patients that had NAFLD compared to the controls. On this slide, you can look at the people that had NAFLD now, and we're comparing F3-4 fibrosis to F0-1 fibrosis. The people with more advanced fibrosis were more likely to have the senescence signature. And if we look across the spectrum of fibrosis in this cohort, we find that the expression of P21 and P16 tends to increase with fibrosis severity. This is RNA, and here's just immunohistochemistry. And you can see this is for P21, more P21 positive hepatocytes in the people with more advanced fibrosis, more P16 positive hepatocytes, and here you see the correlations. So the accumulation of senescent cells would be expected to reduce tissue regenerative capacity. So if the tissue were injured, these cells that are left behind would be less likely to be able to regenerate and replace the cells that died. And so we compare liver regeneration in old mice versus young mice after a partial hepatectomy. This is not earth-shattering. People already knew this, that older animals have decreased regenerative capacity after a partial hepatectomy. But what was important about the experiment that we did is we could ask what's different between the old hepatocytes and the young hepatocytes. So we isolated hepatocytes from old and young animals before. And here I'm showing you at 48 hours after partial hepatectomy. And we performed RNA-seq analysis, and we asked what were the top differentially regulated pathways in the hepatocytes of the old mice, both before and after partial hepatectomy. And what you can see here is that the old mice have reduced expression compared to the young mice of the hedgehog signaling components, and hedgehog pathway signaling was down in the old mice, both before and after a partial hepatectomy, whereas the young animals induced this pathway when the liver was injured and had to regenerate. The old animals were unable to upregulate the pathway. So why am I telling you that? Well, everybody knows I've been interested in hedgehog for a long time, but let me just remind you about the hedgehog pathway. This is a pathway that's developmental, it's active in development, but it becomes reactivated in many cancers, including liver cancer, hepatocellular carcinoma, and cholangiocarcinoma. Hedgehog orchestrates tissue construction by controlling cell proliferation and differentiation, but interestingly, it also controls senescence. In fact, overactivating the pathway in flies and worms is sufficient to increase the longevity of the organism. Also, hedgehog is subject to metabolic control. The ligands and the receptors are directly regulated by fatty acids and cholesterol and their derivatives. In fact, patched and smoothened are cholesterol sensing proteins. So this is a link between fat and metabolism and cell state. And the pathway is subject to metabolic control. What I'm going to show you is that the pathway is not just regulated by lipids, but it actually regulates lipids. And that deleting smoothen is a way that you can get a handle on what this pathway does. So remember, you were holding the thought about what's fat and hepatocytes got to do with premature aging, liver degeneration, and cirrhosis. And hedgehog, which is controlled by fat, controls fat, but it also controls senescence. So what I'm going to show you now is results of some studies where we actually showed this experimentally. So we used AAV8 TBG-CRE to conditionally delete smoothen in healthy adult mice that were on chow. So they did not have a superimposed metabolic stress. And what we found is that within 1 week of deleting smoothen from healthy hepatocytes, we got tremendous increase in liver triglyceride, shown here quantitatively, and here's oil red O staining. Triglycerides went up in the blood, insulin went up in the blood, and the HOMA-IR increased. And when we gave the animals an insulin tolerance test, we showed that there was systemic insulin resistance. Furthermore, disrupting hepatocyte hedgehog signaling in young mice phenocopied the defective regenerative response of the old mice. So remember, I showed you that old mice have inhibited liver regeneration after hepatectomy. Mice that lack smoothen and hepatocytes also have inhibited liver regeneration after a partial hepatectomy. Can you guys still see this?
We can see this one.
Okay, because I lost the other screen. I just want to make sure you can still see.
No, we can see that.
Perfect. All right. So what we've been able to do is we can obviously delete the gene or not in vivo, and then we can isolate the hepatocytes from these animals, and you can see they're loaded with lipid. But their viability, at least upon initial isolation, is not inhibited. However, if we challenge them with something that would further increase oxidative stress, like this GPX4 inhibitor called RSL3, we get some death in the wild type cells, but the rate of killing is much greater in the cells that are lacking smoothing and that are vulnerable. So what these data suggest is that if we block hedgehog in mature adult hepatocytes, they're less able to withstand or recover from metabolic stress. In other words, they're less resilient, suggesting they may be prematurely aged. So to look at that in greater detail, we went to single cell RNA sequencing. So we knocked out smoothing in our mice or not. And then we waited a week when we knew the phenotype was present. We isolated the hepatocytes. We subjected them to single cell RNA sequencing using 10x genomics platforms. I'm not going to go through all of the data. It's been published, but this is our smooth and knockout hepatocyte. And what we were able to show is that that inhibits AMPK, activates mTOR, causes insulin resistance, promotes lipogenesis, inhibits fatty acid oxidation, impairs autophagy, which is why the lipids accumulate in these cells. It also causes lipotoxic stress by enhancing mitochondrial dysfunction, increasing oxidative stress, impairing ATP synthesis, promoting double-stranded DNA breaks, even inducing telomere attrition. So in other words, simply knocking out smoothing within a week caused the hepatocytes to acquire virtually all of the hallmarks of aging. In other words, they became senescent. Around the same time we were doing this work, someone else, Dr. Zhang, published a very beautiful atlas of aging in the mouse, and we were able to compare the signature of our smooth and knockout hepatocytes to the very old mice and found that there were some genes that were similarly shared. But more importantly, if you look at the whole liver, there was a striking correlation between the livers in the smooth and depleted mice and the livers in the very old mice. So I have shown you that senescent cells increase as you go from control to F4. And I've showed you that when smoothing expression falls, you get an increase in senescent cells. So is there a reciprocal relationship between smoothing and NAFL progression in people? And the answer is yes, that smoothing fell in whole liver RNA when we went from control to more advanced fibrosis. And more importantly, the loss of smoothing was associated with worse lipotoxic liver injury. The lower your smoothing, the higher your AST, the higher your NAS score, the worse your ballooning score. So people with worse liver injury, higher AST, NAS, more ballooning, have lower smoothing expression. And then we confirmed the finding in another cohort from Japan. The people, again, that had mild disease had a higher level of smoothing than the people who had more advanced NASH. So hepatocytes with low hedgehog activity accumulate in human NAFL. So what does accumulation of cells that have low smoothing, that are more senescent, have to do with fibrosis? Well, you can see there is sort of at least a reasonable area under the operator curve there, suggesting that there is a correlation between these observations. So how then would these cells that have become senescent, as evidenced by all of those things that I showed you, how would these senescent cells be driving fibrosis? Well, senescent cells have a secretome, and that secretome is thought to be pro-fibrogenic and pro-inflammatory. So we took conditioned medium from our smoothened wild-type cells and our smoothened negative hepatocytes, and then put it on to stellate cell lines, but lots of other different cell lines. Here I'm showing you what happened with the stellate cells. And the conditioned medium from the knockouts were more likely to increase alpha-SMA, collagen, proliferative marker FOXM1. So the conditioned medium from the senescent, smoothened, depleted hepatocytes was more fibrogenic than the conditioned medium from the control hepatocytes. And remember that I had shown you that when we have more senescence, we have more fibrosis. Whoops. So what we're showing is that as the liver disease is progressing, the population of cells that are senescent, in other words, that can't proliferate, is growing. So how do populations of cells that can't proliferate grow? Well, this is a beautiful paper that was published in Nature in October 2017, so a long time ago. It's not about hepatocytes, I don't think, but basically what the authors are showing here is that the trigger for this process is that damage or disease, along with other signals during development, can induce senescence, that these senescent cells have a secretome. They belch out proteins like cytokines, which attract immune cells that are supposed to be clearing these senescent cells. But if for some reason the senescent cells cannot be cleared, they become zombie killers or undead cells. And then these reconfigure the microenvironment. So what I'm going to show you now is that recently a colleague of ours at Duke, Dr. Xiaofan Wang and Chris Penn in his lab, identified a novel signaling pathway that involves thrombomodulin and PAR1 that promotes the viability of senescent cells. And what they found is that when thrombomodulin binds to thrombin and promotes the activation of protein C, that this interacts with this protease activated receptor 1, and it will cleave in a very specific residue. And then at the same time, this complex prevents the degradation of this activated PAR1, and that leads to a sustained activation of survival signaling in senescent cells. So we looked to see whether we could find changes in this thrombomodulin PAR1 signaling access in our human NAFLD livers. And what we found is that, although at the mRNA level, there was not so much of an increase in either thrombomodulin or PAR1, it was significant for thrombomodulin, barely did not make significance for PAR1. We did find an increase in the protein levels. And that makes sense because what this axis is supposed to do is actually stabilize the activated signaling complex. And it's the stabilized signaling complex that's turning on the viability and maintaining the viability of the senescent cells. To reassure ourselves that this was really related to senescence in the liver, we overexpressed P16 using adenoviral vectors in healthy wild-type mice. And as you know, P16 is something that is got to be a very good marker of cellular senescence. And when we overexpress P16 in healthy livers, we get accumulation of lots of senescent cells, so specifically in hepatocytes now. And we see that thrombomodulin and PAR1, which in the healthy liver are combined to sinusoidal cells, become expressed in hepatocytes. And the livers that are overexpressing P16 and have activated this survival pathway that maintains the viability of the senescent cells have inhibited liver regeneration after a partial hepatectomy, as well as after carbon tetrachloride-induced liver injury. So do these non-regenerative senescent hepatocytes promote liver degeneration and cancer? They correlate with it, but are they causal? So the first thing we did to screen for this was to actually induce senescence using a couple of different approaches in both HUH7 cells and primary hepatocytes by either overexpressing P16 or treating them with a senescence-inducing drug. Then we could take the conditioned medium, put it on our bioassay LX2 cells, and show that the senescent medium upregulated the fibrosis markers and the proliferation markers. So it acted like the medium from our smoothen deficient cells that we know are prematurely senescent. Fortunately for us, our collaborators also discovered that there's an FDA-approved drug named Vorpaxar that very selectively blocks the specific activation of PAR1 that results when thrombomodulin and thrombin and activated protein C interact. So this gave us a tool now that we could selectively delete senescent cells by removing the pathway that was maintaining their viability. And I'm just showing you some of the models that we used here. We used a bunch of different models. This is carbon tetrachloride, but we've done it with MCD mice and OB mice and bile duct ligated mice, and the results are very similar. So you can see here we have our profibrogenic agent. And you see that you get a shift of the thrombomodulator PAR1 to the hepatocytes. You also see beta-gal around those same areas. And when we use Vorpaxar, we get rid of these cells that are thrombomodulator and PAR1 positive, beta-gal goes down. But most importantly, we see a regression of fibrosis and inflammation. So we wanted to know, all right, these senescent cells do look like they might be involved in promoting fibrosis. What factors in their secretome might be contributing to that? This is still work in progress, but I'm showing you this one because we and others have shown that hedgehog ligands are very important in activating stellate cells to be fibrogenic. And it turns out that the conditioned medium from the senescent cells is enriched with hedgehog ligands, and the hedgehog ligand biogenesis is increased in the senescent cells. And we've shown that this is biologically active because you can put it on reporter cells and you activate hedgehog signaling activity. And here you can see in our P16 overexpression hepatocytes, upregulation of sonic hedgehog and upregulation of the luciferase reporter gene. Why am I emphasizing this so much? Because a long time ago, Cindy Guy, who works on our group and is one of the NASH serum pathologists, had demonstrated that ballooned or undead hepatocytes that accumulate in the livers of people with NASH strongly express sonic hedgehog. When you get rid of the lipotoxicity with vitamin E, these balloon cells goes away, sonic hedgehog is reduced. Here, when the NASH is active and you have a lot of sonic hedgehog producing hepatocytes, you get a paracrine release of that hedgehog ligand that is activating surrounding stellate cells to become myofibroblastic. When you get rid of the hedgehog producing hepatocytes, you get rid of, or at least reduce significantly, the accumulation of the alpha small positive hedgehog responsive myofibroblasts. So I know you guys are interested in cancer, and I want to circle back by reflecting on this beautiful paper by Lou Roberts and others in Cell in 2017, where they were starting to look at, in hepatocellular carcinoma, the epigenetic footprint of the disease. And if you focus on this side of the box over here, you'll see that there epigenetics here, when they've started to look at microRNAs, the methylomes, the proteome, you start to see hedgehog signaling popping up. And in fact, there's a nice review in Molecular Cancer from 2016 that emphasizes that the hedgehog signaling pathway can affect just about every one of the 10 hallmarks of cancer. The mechanisms are probably different for different things that it's affecting, but it's thought predominantly to be cell autonomous. In other words, that there is paracrine activation of hedgehog signaling that may or may not be affecting the cancer cell itself. If a cell in the microenvironment is making hedgehog ligand, that could be activating the cancer cell, but it could also be that something like a senescent cell is making hedgehog ligand that's activating another cell in the microenvironment that is then making something that feeds back and assures the survival of the cancer cell, and that could be its effects on the immune system. And this is kind of summarized on this slide that again was not focusing on hedgehog particularly, but was looking at general mechanisms by which senescent hepatocyte accumulation could promote liver cancer. Here you see your normal hepatocytes, stress comes in, some of the cells become senescent. These senescent cells are making mediators that would supposedly be recruiting in immune cells to clear the senescent cells. But sometimes the senescent cells can accumulate either because they evade these mechanisms or the mechanisms themselves are somehow defective. And again, just another review, kind of emphasizing that consensus may be emerging here that when you have changes in the epigenome, here again, focusing on methylation, aging, the Horvath clock, that you get changes in the cell populations. This is because some of the things that are normally regulating the epigenome become dysregulated. And then the consequences of that are fibrosis and cancer, just like we've been talking about for the liver today. So the take-home message is that in steatotic liver disease, cirrhosis, and cancer both result from liver degeneration. This is caused by the progressive erosion of hepatocyte functions that are controlled by genetic and epigenetic factors, for example, PNPLA3 and smoothing. This reflects loss of hepatocyte resiliency or biological aging, and that dysfunction precedes and predicts hepatocyte death, deficient regeneration, and progressive organ fibrosis. Dysfunction and wounds that don't heal, cirrhosis and cancer, therefore should be preventable and reversible because biological aging is preventable and reversible. And in fact, although we poo-poo these treatments, they have been shown to modify at least methylation and acetylation of the genome, which is our dynamic maintenance system that regulates gene activity without changing the genetic code. So although these are difficult to enforce, perhaps there will be medication therapy that can have a similar effect. Everybody wants a pill. So maybe novel therapeutic approaches would be to clear the senescent cells using senolytics, or to come up with other ways to reprogram the genome. There are papers already that show that infusions of Yamanaka factors can do this. I showed you earlier about defects of young exosomes. Perhaps you could consider parabiosis with the young form. But I think we should dream big. Immortality is possible. And earlier today, Jin and I were talking about this idea of meiosis, where a senescent cell buds off a piece of itself that then gradually reemerges as almost a stem cell-like population. And that's exactly what happens in this species of jellyfish that is immortal. So I'll stop here and be happy to answer questions. Thanks.
Thank you very much. And that was a fantastic talk. So for whatever technical reason, your camera went off during the talk. So
you couldn't see the slides?
No, we could see the slides, but we couldn't see you.
Yeah, I don't know, because it went off with me too. And that's why I asked you guys if you
No, the slides were fine. Just, if you want
so
to
let me just put it back on. Okay.
Right, that's what I was going to say.
Yeah, well, only the best here at Duke. I have a camera on top of one of my 3 screens here. So it's probably, it's not functioning well. I'm adjusting it now, but anyway, I'm here. It's probably flashing off and on, sorry. But I can see you guys.
Good, okay, so why don't we get started with the, and I just want to take the liberty and just ask a very general, specific question in the beginning, because I remember when I actually introduced you, there was also a lot of alcohol. Yes. Not that I drank alcohol or you had alcohol, but the word alcohol was mentioned several times.
Yep.
And the question I have is, and obviously we all know that alcohol can actually have very similar features than fatty liver disease. So do you see, so how does senescence and alcoholic liver disease, connect with each other?
Yeah, so that's a good question, Tim. And as you know, the real challenge in alcohol-related liver disease is the lack of good animal models.
Don't say it very loud. I think I saw Bin Gao on the call.
Yeah, well, I mean, I think Bin has a great model, but it's very short term, right? And it doesn't really cause a lot of fibrosis or liver cancer. And yet it is well known, as you point out, that actually alcohol-related liver disease is the major cause of cirrhosis and liver cancer. So although it looks like it might be a little bit lower when you look at the relative incidence, because the incidence of NAFLD is increasing and alcohol has always been common. If you look at actually the overall prevalence, really more of that comes from alcohol than non-alcohol related fatty liver disease. But I think the field as a whole has been challenged by the difficulty in getting high penetrant animal models that really get very severe disease. So fortunate for us, we now have big biobanks that are beginning to be assembled that are collecting explanted livers from patients that are undergoing liver transplant for both alcohol-related cirrhosis and severe acute alcoholic hepatitis. We've been fortunate to have funding from NIH. I didn't show this data today, but I am collaborating with my co-PI, Dr. Nash Kalsotra at the University of Illinois, and we've done deep sequencing, bulk RNA-seq, as well as single nuclei RNA-seq on explanted livers of patients with severe acute alcoholic hepatitis and alcohol-related cirrhosis. Many of the things that I've told you today about NAFLD we're also observing in alcohol-related liver disease, but the focus of that proposal is actually looking at RNA splicing, which is not well demonstrated in single nuclei RNA-seq, but we think has a very major role in reprogramming, and we've identified some specific splicing factors that are differentially regulated that actually tend to reprogram the hepatocytes to a more stem-like, if you will, state. What we've been able to show is that not only does the splicing factor expression change, the targets change, the regions of the targets that are differentially spliced, and then correlate that with changes in activity like phosphorylation, nuclear localization, and then targets of the differentially modified target are affected. So we think that there are lots of different levels of regulation, but that's why I'm starting to become a believer in the epigenome. This talked a little bit about methylation because that's thought to be very important in controlling activation of the hedgehog pathway. But we know that there's other things that are also important, one of which would be RNA splicing and microRNAs and other things like that.
Yes.
Dr. Deal, would you mind share the screen so we can all
Yeah,
see?
sure. Unshare the screen?
Yes. So I have
Okay.
Also very generic questions. So we know the the median age of liver cancer, particularly hepatocellular carcinoma in Asia population, but in Africa even more, much younger than those from the Caucasian, from the developed countries. And I don't know, how do you see that the aging-related features are playing a role, particularly in the background of cirrhotic liver disease?
Yeah, I think that's really a great point, Jin. So we know, again, in the United States and probably in most Western countries, that age is a very important risk factor. In fact, one of the major risk factors for both cirrhosis and liver cancer. And in other diseases, I think we have concluded that if age is important, then probably genetic abnormalities are not so important, because it's the people who have the genetic abnormalities that tend to show up with the disease at a younger age. They require less sort of bad living, if you will, to unmask the phenotype. So it's heavily driven by genetics, although one could say that perhaps it's driven by epigenetics, because in those countries there is probably intrauterine exposure to nutrient stress, right? And so we know that could be modified. So they could start off with an epigenome that is old, if you will, because of in utero exposure. So I think it's a really interesting question. I don't know the answer.
Thank you, very interesting.
So, okay, NMAD, it's great talk. I have a question. So your study mainly focus hepatocyte aging and senescence.
Mm-hmm.
So what about other type of cells, like hepatic stellar cells, Kupffer cells, whether they go similar process or?
Sure. So, to be very frank about it, the reason we got into the hepatocyte thing was because it was supposed to be a negative control that turned out to be not negative. So as you know, my lab has for a long time studied how the hedgehog pathway activation is very important in causing quiescent stellate cells to be myofibroblastic, in switching the immunophenotype of macrophages and NK cells, how it affects cholangiocyte proliferation. So we were saying that activation of the hedgehog pathway is a bad thing, and it is if it's overly activated. But we had these ability to manipulate the pathway in hepatocytes, and we thought the pathway was actually not important in hepatocytes. So it was supposed to be a negative control. So we gave the AAV8 TBJ-CRE vectors to the smooth and flox mice, and when we came back a week later, everything was supposed to be normal, except it wasn't. When we opened the abdomen, the liver was fatty. And after I accused people of giving the mice the wrong virus and everything else, it was clear that it was a real phenotype. And so it was so striking, we decided to pursue that, and I showed you what we got. That does not mean that overactivation of the hedgehog pathway is also bad. So it's kind of, you have to have a sweet spot. And we think in liver cancer, in fact, that the hedgehog pathway is overly activated. And the issue is, why does that occur? But the point is that these pathways need to be present in the right amount at the right place in the right time. So to answer your question about other cell types, yes, we have looked extensively in, for example, hepatic stellate cells. We just published a paper, I think it was in maybe gastroenterology, I'm not sure, it could be hepatology. But anyway, where we manipulated the YAP pathway in stellate cells and basically showed that senescent stellate cells are less fibrogenic than myofibroblast stellate cells, that they are susceptible to different kinds of death signaling, and that this process is heavily controlled by the Hippo-YAP pathway. And then we could conditionally delete that pathway in myofibroblastic stellate cells in vivo and reverse fibrosis and improve liver injury. So clearly there are lots of different cell types that are experiencing these things. And I think that we are all en route to some exciting discovery now that we have the ability to do single cell RNA sequencing, spatial transcriptomics, all those kind of things to really start to tease apart what is it in the microenvironment that is making cells that seem to be very similar behave very differently. And I think this whole miracle of plasticity and all is kind of where disease is. And so we're just beginning to tiptoe into that quagmire.
Thank you.
Hey, Wynn.
Yeah, Anna Marie, great to see you and
Thank you.
Hear you present your exciting work, which I've been reading when it's in the various publications. I wonder whether in these senescent cells, is there a role for the LKB1 AMPK pathway in the sense that that's involved in PAR1 phosphorylation and multiple downstream effects, ranging from the mitochondria to polarity to big issues at the cell biologic level. So is there any evidence that the AMPK LKB1 pathway participates in the effects that you've observed?
Yeah, perhaps I didn't have time to explain it in detail, but we recently published a paper about that. So many years ago, more than 5 for sure, some workers in Europe were studying obesity in model organisms. And they did tissue-specific knockouts that were conditional in flies. And what they ended up concluding was that the hedgehog pathway was the single most important regulator of total body energy homeostasis in flies. And then they went ahead and made mammalian models in mice and confirmed that hedgehog critically regulates adipogenesis. That same group was then trying to understand the mechanisms for that. And they came across they discovered that smoothen regulates AMPK activity. And if you don't have smoothen, you can't activate AMPK. And we confirmed that. We had, at the same time, been working in stellate cells and come to a very similar conclusion that smoothen was necessary for AMPK activity. Now, the workers in Europe had linked it to the pathway you talk about, the LKB1. We have not done that. However, in these studies that I was describing to you today, what we think is that a central mediator of the process is actually the non-canonical hedgehog signaling that leads to the activation of AMPK. And in fact, I didn't show you today, but we can reverse the phenotype by using agonists of AMPK. So in the absence of AMPK, what we get are changes in autophagy. So AMPK is supposed to inhibit mTOR. So mTOR is overly active. If you have overactivation of mTOR, you have decreased autophagy. If you have decreased autophagy, you accumulate lipids. It's also one of the things that's thought to be promoting senescence. The changes in mTOR, we also linked to changes in FOXO1 phosphorylation and changes in gluconeogenic gene expression, and therefore insulin resistance. If you inject these animals or cells with insulin, they do not phosphorylate the insulin receptor. There's tremendous changes that are regulating signaling when you take out smoothened. And we think that one of the central nodes, the two key things that we've been able to identify are AMPK and mTOR. And we think that they are interrelated. So although there are effects on the GLI transcription factors, we think this is being actually mediated mostly through the metabolic effects of the pathway, which are already known to be regulated by AMPK. So yes, we think AMPK is very important.
Okay, so there's one question from the chat, Diana Dinowitz. Maybe you want to ask yourself before I read this?
Okay, thank you, Dr. Deal. That was a very interesting presentation. I was wondering how environmental exposures with transgenerational effects can influence the likelihood of cirrhosis and cancer in future generations. And then like a part 2 to that question is, what can we do to mitigate that if it's possible?
Right, so I think you raised a very important point, and this is not our work, but you may be aware of work that came out of the group from the UK, which showed that protection from cirrhosis could be inherited. So what that group did is they treated male mice with carbon tetrachloride, got cirrhosis, withdrew the carbon tetrachloride, the cirrhosis regressed. Then they bred the grandfather mice and got their sons, and then exposed them to carbon tet, took it off, let them recover, bred them, and got the grandson. So the F2 generation, when exposed to carbon tetrachloride, was actually protected from carbon tetrachloride-induced fibrosis. As far as I know, they haven't yet worked out the mechanism for that. But as I said earlier, we know that in utero, exposure to metabolic stress, either over or under nutrition, has profound effects on the epigenome. So, but I don't know, maybe somebody else on the call does, if anybody has really been able to define the mechanism whereby that might be affecting these pathways that we've been talking about, or cirrhosis. And yet we do know that 50% of the chance of getting cirrhosis in NAFLD is inherited. We've identified, as we talked a little earlier, several genes. The field has identified, not me, of course, but genes that are associated with an increased likelihood of getting cirrhosis and cancer. But we know that those genes alone are not sufficient to cause the phenotype, because the phenotype really blossoms when you expose the individual to metabolic stress. So, complicated way of saying that there's probably genetic and epigenetic interaction. In utero exposures are likely to be important, do seem to be important, particularly in NAFLD. But I don't know if anyone has ever figured out the mechanisms for that. Does anyone else know? I don't. But somebody should do research on that.
All right, so we have, I think, less than a minute left. Natalie, you have a very quick question.
Yes, just if you can elaborate how hedgehog is related to liver zonation and whether you think that the degeneration occurs in a non-uniform manner. Do you know? Have you looked at that?
Yeah, so we do have we published a couple of papers using single cell approaches to look at that. So I'm sure you're aware of work from Hao Zhu that showed that if you kill zone 3 hepatocytes, zone 2 hepatocytes can replace it, right? And if you kill zone 1, you get zone 2. Interestingly, our single cell analysis suggests that the most smooth and active subpopulation is in zone 2. We also looked after partial hepatectomy in mice and identified cells using RNA velocity kind of approaches that looked like they were being reprogrammed to be more fetal-like. In those reprogramming populations, we could identify pathways that were differentially activated or repressed. Hedgehog was one of the differentially activated pathways. And then by using ATAC-seq in the same animals or same samples, I guess, we could identify that binding sites for hedgehog regulated transcription factors became more accessible in the cells that were being reprogrammed to be more fetal-like, and those are the cells that are more proliferative. So we think that the hedgehog enriched population, or at least the smooth and active enriched population, is in zone 2, and that after a partial hepatectomy, I don't know whether it's only cells in zone 2, but cells get reprogrammed to activate that signaling pathway, or at least some of them do. And then the cells that actually reprogram open up their chromatin, and their accessibility to GLI binding factors is increased, among other things. So that's not the only thing, for sure.
All right. Thank you so much, Anna-May, again.
You're welcome.
It was a fantastic talk.
Thank you. Bye, guys.
Bye-bye.
Thank you.
Bye.